So this is genetics. This is SC912L16.1. Genetics is another one of my favorites. You can see everything online, and don't forget to check your dates. This one involves a lot of practice. If you don't practice, you're not going to get it. I'm sorry. This time you're going to have quite a few vocabulary words. They are all going to be tested at the same time because they're that important. Your vocabulary words are allele, codominance, dominant, F1 generation, F2 generation, genetics, genotype, heredity, heterozygous, homozygous, incomplete dominance, law of independent assortment, law of segregation, monohybrid cross, multiple alleles, P generation, pedigree, phenotype, polygenic inheritance, probability, Punnett square, recessive, sex-linked gene, test cross, and true breeding. So heredity. What is heredity? Heredity is the passing of characteristics from parent to offspring. So that's the passing of whatever your parents have onto you. You're considered their offspring. So you might think it's a random occurrence that some of these things happen, but these are actually traits that you pass, that you inherit from your parents. So the hitchhiker's thumb is one. So you can bend your thumb backwards, or does it sit up straight? Your earlobes, whether they're attached or not attached. The widow's peak, that's there at your hair, where kind of like at the center of your head. The rolled tongue, whether you can roll it or not roll it, whether you're right-handed or left-handed. Your mid-digit hair, that is in between your fingers, whether you have that or not. Freckles, dimples, the cleft chin is another one. Whether you clasp your hands left over right or right over left. Whether or not you have 6 fingers. Whether your pinky's bent or does it sit up straight, meaning the top part of your pinky, sometimes it bends. And then whether you have a longer big toe. So is your big toe longer than the second one, or is it shorter? Those are all things that are passed from our parents. We might think that it just happens just because, or we practice so hard. But no, those are actual things that have been passed down to us. The father of genetics that's Gregor Mendel. He's a name that you probably heard before because he's so common. He was a monk who studied pea plants. But why pea plants? Pea plants have a variety of traits, and they only fall under one allele, which you'll learn about later. So their traits are the flower color, the seed shape, the pea color, the plant height. They are easy to obtain and breed. They have a short generation time. They produce a large number of offsprings. And then we have what is genetics. Genetics is a branch of biology that studies heredity. People once believed when we were born that the traits that we came about having were just a blend of our parents. Meaning if one parent was tall and the other one was short, we're going to be medium. It later became known that obviously this was not true. So in order to find out that this was not true, Mendel concluded that for each inherited characteristic, an individual has 2 copies of the gene, one from each parent. There are alternative versions of the genes. These are called alleles. You get one allele from each parent. When 2 different alleles occur together, one of them may be completely expressed, while the other might not have any observable effect on the organism's appearance. The dominant trait is the one that is expressed. It's dominant, like the dominant person in a relationship. It's represented by a capital letter. The recessive trait is masked, meaning it's not shown, and it's represented by a lowercase letter. Just because it's masked doesn't mean that it's not always shown. It's going to be shown at some point, and we'll learn about that. So how can that recessive allele actually be expressed? Well, in order for the recessive one to be seen, for you to see, let's say, that person in the relationship, there's got to be 2 of them. So the only way that you're going to see the recessive allele is if there are 2 lowercase letters. The other one, dominant, it only takes 1. Yes, you can have 2 dominant things in the, you can have 2 capital letters, but it only takes 1 for you to see it. Now, there are some exceptions to this rule, of course, but for the time being, it only takes 1 for you to see the dominant allele, or the dominant trait. There are some keywords that you need to become familiar with because you're going to be using these as you answer questions, and they'll be used in regular language spoken in this class. Those words are homozygous, heterozygous, genotype, and phenotype. Homozygous is both alleles for a trait are the same, meaning, you all know homo. If you call somebody homo, that means they're the same, they're with the same type of person. So in this case, it's going to be 2 capital letters or 2 lowercase letters. Heterozygous means different. That means you're going to have 1 capital and 1 lowercase, 1 recessive or 1 dominant. A genotype is a set of alleles for the trait. So the genotype is what actually makes you up. So you're either going to have big T, little t, or big T, little t. You're either going to be homozygous or recessive, or homozygous recessive, homozygous dominant, or heterozygous. Phenotype, though, that is actually what we see, what is expressed. So that is your physical appearance. So big T might stand for tall. So for your phenotype, we're going to see that you're tall. We're not going to see that you're big T. You can be tall, short, green, whatever. Some phenotypes are influenced by the environment. For example, I can lay out in the sun and I'm going to show up with a lot of freckles. That's part of the environment, the sun. Your muscle size, you can go to the gym and work out and your parents can be as tiny as possible, but because you work out so much, you're going to develop muscles. That's something that is influenced by the environment. These are all things that can affect your phenotype. So we have your mama and we have your daddy, but how do we know what you're going to end up getting? Well, in order to find this out, we have to use something called a Punnett square. A Punnett square is a diagram that predicts the outcome of a genetic cross by considering all possible combinations of gametes in the cross. It's 4 boxes inside of a square. This represents a monohybrid cross. Or if you have 16 boxes, it's called a dihybrid cross. The possible gametes from one parent are written across the top and the other one's written down the side. Punnett squares are really easy to do, but they do take a lot of practice. I've attached a lot of practice for you to work on. You'll be required to work on, but please make sure you are practicing the Punnett squares. Here are some different examples of some crosses that can be done in a Punnett square. The first one is a homozygous dominant, meaning it's 2 capital letters, and they're the same, and a homozygous recessive, which is 2 lowercase letters. So what they've done is they've wrote the mom across the top, and they wrote the dad down the side. The dad in this case is big A, and the mom is little a. So as you can tell, they've taken them and they wrote them across the top, down the side, and then they fill in the middle part. So in order to fill out the middle part, you basically can take your fingers and pull down from the top, over from the side, and that's what's going in the first box. Move over to the second one. Pull down from the top, over from the side, and that's what you're getting there. Then you have to determine actually what you have. In this case, they're all big A, little a, which means that they're all heterozygous. Later on, you'll find out that this is a phenotypic ratio of 4:0, and it's a genotypic ratio of 4:0. Then we have the other one. They did heterozygous. Heterozygous means different. So both the mom and the dad are big A, little a. So they ended up crossing them. Again, they pull from the top, pull from the side, and we come up with something a little bit different. This genotypic ratio is a 3:1, meaning we have 3 of these giraffes that are going to represent the dominant gene and 1 that's going to represent the recessive. In the phenotypic ratio, remember that's what we see, we're going to have a 1 to 2:1. We have 1 big A, 2 big A, little a's, and 1 little a. Again, you will have lots of practice on this. There are 3 major steps in the crosses. The first step is known as the P generation, or better yet known as your parents. This is each variety self-pollinates for several generations. It ensured that each was true breeding. True breeding means that they're definitely homozygous dominant or definitely homozygous recessive. True breeding is when all the offspring display only one form of the character, meaning it's purebred. You've heard of purebred dogs. That's what we all aim for. We want a purebred animal. Purple times purple gave them purple. White times white gave them white. That's what they want. That's what we want when we choose our animals. Step 2 is referred to as the F1 generation. So this is the first generation to follow from the parents. This is when you cross-pollinate two pea generation plants with contrasting traits, meaning that we're going to cross that purple with that white flower. The ratio is always going to be a 4-0, meaning you're always going to see the purple. So it's going to result in 100% purple flowers. Why would no white show up? Well, in order to figure this out, I'm going to do a Punnett square for you on the board to show you the exact way. The F2 generation is just a cross between the F1 generation. They're going to self-pollinate. This is always going to result in a 3-to-1 ratio, and again, I'll give you an example on the board. So the laws. There's two laws that we talk about when we talk about genetics. It's the law of segregation and the law of independent assortment. The law of segregation basically just states that things are going to segregate. They're going to separate. The two alleles for a character separate when gametes are formed. Gametes are sex cells. So the sperm and the egg, they're going to separate. The law of independent assortment is when you use a dihybrid cross, which is a 16-square Punnett square. It found that the inheritance of one character did not influence the inheritance of any other character. For example, plant height does not influence flower color. Now there are some, though, that do influence each other. Hidden traits. Hidden traits are something like you bought this dog and you know it's a lab. And they told you it's a lab. You don't have paperwork on it, but they told you it's a lab. Well, all of a sudden you cross it with another lab and you end up with something a little bit funny. It's not exactly what you thought it was going to be. This is known as a hidden trait. In other words, can you tell by looking at that purple pea flower, wherever you are, if they are true breeding for the trait, meaning are they a purebred for that trait? No, you cannot. You cannot look at something and tell that it's true breeding or not. You can't see inside of me whether I'm true breeding for brown hair. You don't know what my genetic makeup is. How does crossbreeding help determine if a plant is true breeding for a trait? So how is crossbreeding going to help to determine that one day me and Jamie has brown hair, are going to have kids with brown hair? How's this going to work? So in order to find out if something is true breeding, whether if it's purebred, we have to perform something called a test cross. This is a test that is used to determine an individual's genotype. Genotype is what we're made up of. You cannot see it. You're going to take an individual whose phenotype is what we can see, is dominant, and cross it with a homozygous recessive individual. So we know that purple is dominant for pea plants, and white we know is recessive, thanks to Mendel. So if we were to take a white pea plant, because we know its genotype is little r, and cross it with a purple one, who we don't know whether if it's big R or big R little r, we can be able to find out. This is called a test cross. The phenotype of the offspring will allow you to tell the genotype of the first individual. So, if we get any white flowers in this cross, we know that that purple flower had to have a little r in it because it had to be heterozygous, because in order to get a white flower, we have to have two little r's. We can't see it with just one. If we get all purple flowers, then we know that that purple pea plant was a purebred, homozygous, dominant individual. Okay, so if you couldn't tell already, this is the test cross situation that I have set up. I've completely imagined on my own, and we're going to perform this test cross. So, with pigs, I know that straight tails are dominant over curly tails. No, I don't really know that. But for the situation, straight tails are dominant over curly tails. One day, Farmer Jamie went into the pen and found that he had a litter of 10 piglets, but he doesn't know who the father is. What he did know, though, was that the mama sow had a curly tail. Sow is known for a pig who has had babies. Can he determine the genotype of the father by looking at the piglets' tails? How? How might he be able to do this?